The overall goal of the following experiment is to measure mRNA decay rates in the yeast Saccharomyces cerevisiae. This is accomplished by first growing yeast cells to log phase at the permissive temperature. Next, the cells are shifted to a higher temperature in which total mRNA synthesis is inhibited. Yeast samples are then harvested at predetermined time points. For each sample, the total RNA is extracted, separated by gel electrophoresis, and transferred to a membrane. Finally, northern hybridization is performed to ascertain any changes in mRNA levels as a function of time. Ultimately, this protocol can be used to measure mRNA decay rates of individual mRNAs or genome-wide in different growth conditions.
This method can help answer key questions in the RNA decay field, such as, do RNA decay rates vary in different cellular conditions and genetic backgrounds? Demonstrating this procedure will be Megan Peccarelli, a graduate student from my lab.
To begin, prepare a starter culture by inoculating a single yeast colony into 5 milliliters of an appropriate selective media for the strain under consideration. If the strain harbors a temperature-sensitive allele of RNA polymerase II, grow the cells to saturation overnight at a permissive temperature of 28 degrees Celsius. On the next day, set up a series of larger overnight cultures, each at a total volume of 100 to 150 milliliters.